a nerve can be stimulated. Okay? The number 2, the all or nothing rule. You might be having a hint on this, or you might not, but we are going to see what exactly it is about. Then the transmission speed. Okay? What are the factors that will affect the speed at which a given impulse is going to be transmitted? Okay? And one of the factors is the myelin sheath and the axon diameter. So these ones, they do affect the transmission speed of a nerve impulse, of an electrical impulse. And so we should be in position to explain briefly and extensively how these two, axon diameter and the myelin sheath, how they do affect the transmission speed of an impulse. Then we shall also talk about summation, okay, as a concept, and we see what it is about and how does it bring about. Okay, yeah, that's pretty good. Sorry. Now with the stimulation, this one is simple, how an impulse can easily be evoked, how a nerve or how a neuron can be stimulated, how it can be excited. So most of the times, impulse are a result of excitation of receptors. Okay? So most of the receptors that you have, when they get stimulated by a stimulus, they undergo what we call excitation. Okay? Where they either get depolarized or they get hyperpolarized. And so that one is enough to excite a receptor to produce a nerve impulse which evoked and transmitted to its destination. However, that is not restricted to receptors alone. Even the axons, an axon can also be stimulated, or it can also be excited. Okay? So however, if the exact, or if it is excited by the exact direct application of an appropriate stimulus, then you can cause depolarization within an axon. For example, if this is your axon, okay, and we are used that this axon is just for transmission, okay, of the electrical impulse. However, the same impulse that excited the receptor, if it is applied to the same axon here, this axon can be excited and a wave of depolarization can be created and transmitted along this axon. Okay, so it is not just restricted to receptors alone. So usually nerves are stimulated by mechanical, osmotic, chemical, thermal, and electric stimulus. And these are the stimuli that are within our surroundings. If it is not mechanical, for example, touch, then it is heat, then it is chemical. For example, somebody might put on a deodorant and you are stimulated. For example, if you are hypersensitive, if you are allergic, then you respond to it. Or it could be an electrical stimulus, depending. Okay? Yes. So in case you have a question, you can feel free to drop it in the chat box. Now with the all or nothing rule, this one is just simple. It is either or nothing. Don't make life hard. It is either or nothing. So from that, what comes into your mind? It's either or nothing. If I want to eat and you have the food, you either give me the whole of it, a full-sized plate, so that I eat and get satisfied, or you don't, you stay with your food, but not just giving me pieces, pennies. No. Okay? It's all or nothing. It's like right now, fees. We are paying 30 a week. So all or nothing. You either pay the 30, that is if you want to pay, or you don't pay anything. So that is all or nothing. And basically that's what it is. So it states that if the strength of the stimulus is below a certain threshold value. Now this is just an English word, okay? But we use it also, we borrow it. That this value, it is the minimum value required for an action potential to be evoked. The minimum value, the minimum intensity of the stimulus that is required for an action potential to be evoked. Now, if the strength of that stimulus is below that value, then no impulse is going to be evoked. No action potential is going to be evoked. But however, if now the strength of the stimulus is just equal or above the threshold value, then a full-sized potential is evoked. And this full-sized potential will always remain the same, however much the intensity of the stimulus is increased. So basically, that's how it is simple. That's how simple it is, and it can be illustrated using those two curves. You can see here it is our resting potential, and you have your stimulus here. Let me use some different pen. So you have your stimulus here, okay? Oh, now that one is worse. So you have your stimulus here, okay? However, if the stimulus is weak, these graded potentials, they want to reach a given value, okay? They want to reach a given value. By the way, this question is specific. This question is good. It can come in any paper, okay? It can come in any paper, so take care. So if maybe, let me rub it again, and maybe you are not following. If this is my stimuli, and this is some small stimulus, okay, and this is some small stimulus, so if these stimuli are not small enough to reach this value, then no action potential will be evoked. So the wave of depolarization will always build up, but it won't reach the threshold value, so you won't see anything. It won't be interpreted. No signal will be sent to the brain. However, if you keep on increasing the stimulus, okay? If you keep on increasing the stimulus here, okay? And you finally reach that stimulus that is enough, okay, to evoke an action potential to reach the threshold value, okay, then a full-sized, like how you see, okay, a full-sized action potential is going to be evoked. That even if you increase the stimulus like how, it will always remain the same full-sized action potential. Okay? So as long as the stimulus is above the threshold value, then is equal or above the threshold value, then a full-sized action potential is evoked. So that's why it is called or. Or why? Because full-sized. Or nothing. Why nothing? Why nothing? Because the intensity is way below the threshold value. Is that okay? That is basically how it is. That is basically how it is. And you should be in position to explain that fully. Yes, now checking for the chat box so that I can attend to your questions. No questions yet? Why? Please drop your questions. Feel free to drop your questions. So that is basically what we have to talk about, the all or nothing rule. Now another property, or the properties of these nerves, also depend on the refractory period. Now the other time we talked about the refractory period, but today let's talk about it again. Now this is the period of inexcitability, the period where a neuron, a nerve, or a receptor cannot be excited. You cannot excite that neuron. Okay? Yes. Why? Because that neuron is under the recovery phase. Okay? It is recovering. It has conducted an impulse, but finally it is in that recovery phase. It is trying to restore back the resting potential, the membrane potential. So in the process, as you are trying to recover the membrane potential, then however much the impulse is strong, and we shall see the types, then you cannot excite that impulse, that neuron, that receptor again. Okay? So since you cannot excite that receptor again, then we shall say that the receptor is in refractory period. Okay? Yes. So we can categorize this refractory period into two. And the first one is the absolute refractory period, where in this one, okay, you cannot excite a neuron, you cannot excite a nerve, okay, or a receptor, however much the stimulus is strong, like how, okay. Why? Because it is that period that is just immediate the disturbances. Okay, so we have said that if this is your axon, okay, and we shall find a graph down. If this is your axon, the inside will always be negative and the outside will always be positive. But when an action potential, when an impulse arrives, it reverses and the inside becomes positive, okay, and the inside becomes positive and the outside becomes negative. So immediately, as soon as this point, as soon as you form this, okay, that point that is immediate, you cannot excite, you cannot re-excite a nerve fiber or a receptor again before that time reaches. Okay? Because already the inside is positive. You have to first restore back the resting potential, the membrane potential. Okay? To prepare it to receive some impulse. So it is a period that immediately follows the firing of a nerve fiber, where an axon is completely incapable of transmitting a nerve impulse. Then another one is relative refractory period. And with this one, here there is some excitability, that is, if the stimulus is strong enough. Okay? So it is a period shortly after firing a nerve fiber, where there is partial polarization has occurred. Okay, after partial polarization has occurred, and there is possible to generate, or it is possible to generate an impulse provided the stimulus is longer than normal. Okay? So here, if you continue stimulating, you can end up evoking a nerve impulse. So these two, they are refractory periods, where one is absolute, another one is relative. So from that graph, you can tell. So here, this is your nerve impulse you have evoked, and you have really depolarized, and I hope you should be in position to describe this curve. Okay, you have really depolarized. Now, shortly after this, let me change, let me get some pen. Now, at this stage here, okay, they are the same with the graph up to here. Okay, you cannot excite this nerve fiber again. You cannot excite it. Okay, why? Because it has been totally disturbed. So however, it is believed it is from here to here, because from this point, that's where you have your stimulus, okay, and it is trying to build up to the action potential. So between this period, you cannot excite this neuron, however strong, however long lasting a stimulus is. So this one is a refractory period, and it is an absolute refractory period. However, from here, between 1 up to around here, where you have completely restored, because you can see that the resting potential is this. So, up to somewhere here where you have completely restored. Okay? So the whole of this is hyperpolarization. Okay? And here you are trying to restore the membrane potential by pumping sodium ions out and pumping in potassium ions. So this period, the whole of this, you cannot excite a neuron. However, if the stimulus is high and longer than normal, then it can end up exciting, because it might end, you might restore your membrane potential when the stimulus is still there. Okay, so that you excite your nerve, your neuron again. So basically, that's how it is simple. It is as simple as that. Now I'm checking your chat box. So before the chat box, we can talk about the importances. Now, is this important? Yes, it is actually so important. Okay? By these 3 reasons. And the first one is, it determines the maximum frequency at which an axon can be, so they can transmit an impulse. Because you cannot keep on transmitting an impulse. Okay, and you can't transmit at a high speed, like you continuous stimulation. Okay, it can end up damaging the brain cells, which leads to trauma. Okay, in fact, you can read about how pesticides work. Okay, yes, we are going to study the synapse, and I'll talk about it. So, but this refractory period, it determines the maximum frequency at which an axon can transmit an impulse. Okay, so that you don't exaggerate things, and if you exaggerate, then you overreact. Okay, and it might not be good for you. It ensures separation of action potentials and specify the stimulus that is causing an excitation. Yes, imagine if you just excite continuously. How are you going to respond? How are you going to respond? So it separates the different action potentials, okay, that are stimulating a particular neuron, and then determines how a body or how the brain is going to respond to such a response. Then finally, which is the most key, it prevents spreading of action potentials. And I think I talked about this the other time. It prevents spreading of action potentials, which prevents the backflow of a chemical impulse, of an electrical impulse. For example, if maybe this is the direction of your nerve impulse, okay? and you have created a wave of depolarization here, okay, in this region. The question is, they are just ions. Hmm? It is just a wave which just moves. Actually, the good illustration is, if this is a water body, okay, this is a water body, and you throw a stone here, isn't a wave going to distribute just randomly in among all sides? Yes, it will just spread through all sides. So the same applies to this. It is just a wave of depolarization. Now, what maintains its unidirectional flow? It is because when you stimulate this region, okay, then there is no way how you will stimulate this region back. And so if you can't stimulate this region back, then this region will be safe. So as a result, you keep on stimulating this side, this side, this side, this side, this side, until when you reach the brain, and the brain also sends until when you reach a target organ. Is that so? So it maintains a unidirectional flow of an electrical impulse, so that it doesn't create confusion, perturbances within the speed at which. Somebody has something to say? Somebody has something to say? Okay, seems no one. So now the transmission speed, this one, it is the speed at which a nerve impulse is going to be conducted along an axon. Okay? The speed at which a nerve impulse is transmitted. Now it is determined by two factors, and you should be in position to explain how these two factors affect the transmission speed. Okay? Now it depends on the type of a neuron of an animal in question. And the variation depends on, now depending on the neuron, okay, it will determine the transmission speed, it will determine the speed at which a nerve impulse is going to be transmitted. And the first one is the axon diameter. Okay, so we have different axon diameters, where some animals have their axons with narrow diameters, and others, such as a giant squid, has axons with wide, okay, diameter. So now this difference in axon diameter determines the transmission speed. Okay, the speed at which an electrical impulse is going to be transmitted along these two axons is going to be different. Okay, as we are going to see. Then the next one is whether the axon is myelinated or non-myelinated. Now this one comes from myelin sheath. Don't get confused. Myelin sheath. Okay, and so if this is your axon, okay, so some axon are myelinated, where they do have the myelin sheath, okay, and others they do not have, they don't have the myelin sheath. So now this the myelin sheath also affects the transmission speed, as we are going to see. Now with myelin sheath, let's use this. If you have the myelin sheath on your axon, then, because remember we agreed that the myelin sheath is made up of fat, it is just fats that are secreted by the Schwann cell, okay, Schwann cell. Now these fats, they do insulate, okay, they do insulate exchange of ions, the potassium and sodium ions, across the membrane. that this point that has the myelin sheath, you cannot have any ions crossing there. Okay, this point that has myelin sheath, you cannot have ions crossing there. So what does that mean? It means that if the ions cannot cross this step where you have the myelin sheath, then the wave of depolarization will always occur in the nodes. Okay, in the nodes. Let me draw something here. That's the myelin sheath. That's another one. That is another one. So the same applies here. Myelin sheath, myelin sheath, myelin sheath. So the wave of depolarization will always occur, okay, in these points that do not have the myelin sheath. So that's why you'll be having depolarization, okay? But these points with myelin sheath, okay, you won't have there any depolarization. Now somebody is thinking about this being disadvantageous. No, it is highly advantageous that the action potential or the wave of depolarization is going to occur in form of jumping, or you can say leaping. It leaps from one node of Ranvier to another node of Ranvier, to another node of Ranvier, to another node, to another node, to another node, to another node. Okay? And this node, this mode of impulse transmission is the one that we call the saltatory impulse transmission. Saltatory means jumping, leaping. Okay? So it is saltatory impulse transmission. And it is quick, that you just depolarize these parts, okay? The nodes of Ranvier. And you don't depolarize these parts covered with the myelin sheath. So as a result, the impulse is going to be transmitted at a relatively high speed than when this axon was non-myelinated. That if it is non-myelinated, for example, it will just be uniform. The wave of depolarization will just be uniform. Okay? You depolarize this membrane uniformly. And so this one is slow compared to when you depolarize in the form of leaps, jump. Is that okay? Yes, that's pretty good. Now another factor which affects the transmission speed is axon diameter. Now this is how wide an axon is. And the wider the axon, okay, the faster the impulse transmission. We can say that thicker the axon, the faster the impulse transmission. Why? Because if the axon is wide enough, then there won't be ionic interferences. The sodium ions, the potassium ions, the chloride ions where possible, they won't interfere. Okay? And so because of that, it will create a large surface area over which the ions will easily be exchanged between the outside and the inside. And so as a result, you will easily depolarize a neuron or a membrane. Okay? And so some invertebrates such as the squid, the annelids, the crayfish, and the crustaceans, their axons have a wider diameter. Why? Because they need to respond to their surroundings. They are easily predated on. So they must be able to quickly respond than how their predators do respond to enable them survive their surrounding. So basically it is as simple as that. So before we talk about the synapse, let's check if there are questions. We attend to the questions. Any questions for us? Any questions? No questions for me? So should I assume everything is understood? Which is not a good assumption. Okay, so let's proceed. Now with the synapse, if you remember our introductory part, that drawing, that structure, that picture that I put in the background, it was a synapse. And what is it? It is that point, okay, where a given neuron, a given axon terminates to another axon or to another neuron. And it is simple. So an action potential that is passing down an axon eventually reaches the end of an axon, okay, and all of its branches. So if this is your axon, maybe that is the cell body, okay, cell body, and then there is an axon, there is an axon. Now this axon, it will terminate into branches called dendrites. So very many of them, okay. Now the axon potential that is traveling through this axon, it will move until when it reaches the very end, okay, and it must be transmitted to another axon, okay, maybe that is a dendrite, then the cell body, and then finally transmitted to another neuron. So this point of connection is what we call a synapse. Let us proceed. Oh, sorry. So branches may form junctions, okay? These branches, the dendrites, they may form junctions with the dendrites of other neurons, or with muscle cells, or with glands. So depending on a given neuron, if it is a motor neuron, then obviously it will form junctions with the muscle cells or with the glands. But if it is a sensory neuron, then it will obviously form junctions with other neurons. Now, such junctions are called synapses. Okay? Such intercellular junctions are called synapses. Now, this synapse, it has some good science that no one should miss. Okay? That all of us should know. And the science is what we are going to talk about today. Okay? Yes, that's good. So the structure of a synapse, in case they tell you to describe a synapse for us, what are you going to talk about? Or what should I expect in your essay? Now in your essay, number one, there should be, you should tell us that it is a swelling. It is a swelling because you can see there's always some swelling because before that one connects and another swelling, okay, before this one connects. So you are going to see the structure here, don't worry. So it is a swelling at the end of a nerve fiber, okay, called a synaptic knob. Now there are two types of synaptic knobs, where you have the presynaptic knob and the postsynaptic knob. Now, pre and po. Pre and post. Pre means before, post means after. So that knob that is between this neuron that brings the impulse becomes the presynaptic knob. But this one that now takes the impulse from this neuron to this neuron is called a postsynaptic knob. Is that okay? Yes. Now inside The presynaptic knob, okay, consist of numerous mitochondria. Now, what are these mitochondria for? Transmission of energy. Okay, so they provide energy. And what is the energy for? Impulse transmission. Because remember, we talked about that the transmission of impulses involve exchange of sodium and potassium ions. Okay, now after you've exchanged, you need energy to restore back the resting potential. Now, where are you going to get that energy from? The mitochondria. Now you should be thinking about adaptations. Okay, why is it even swelling? Think about that. Why does it have mitochondria? Why does it have the synaptic vesicles? Now these synaptic vesicles, they are the ones that do contain our neurotransmitter substance. They are just vesicles, just a sac, okay, with the neurotransmitter substance inside, okay. Now, if you remember our signaling pathways the other time, if you remember, we talked about the neurotransmission, that one which occurs within the two neurons. And one of the examples we gave, the neurotransmitter, e.g. acetylcholine. And I briefly talked about how the impulse can be evoked from one neuron, from the synaptic knob, presynaptic knob to the post. Okay? But today we are going to talk about it in the details. So you have there the synaptic vesicles. Now these ones, they do contain a neurotransmitter substance. And it is this neurotransmitter substance that plays an important role as far as the impulse is going to be transmitted from the presynaptic neuron to the postsynaptic neuron. Is that okay? Yes. So I expect you to talk about the neurotransmitter substance. And we have different neurotransmitter substances. But the common one is then choline. Oh my god, sorry for that. Acetylcholine. So that is the most common one. But there are very many. And each neurotransmitter substance has its own effects. Let me first moisten my hands so that I can write well on the board. Okay, that's pretty good. So I also expect you to talk about, you talked about the neurotransmitter substance and its role. And finally, you talk about the membranes, the presynaptic and the postsynaptic membranes. And finally, you draw. Before the drawing, there is that space. Because these ones, they don't just connect to each other. No, they form some space. Okay? And actually, this space is important. It is where the activity of this synapse occurs. Okay? To allow the impulse to be transmitted through the synapse. And the drawing is here. Okay? So let me clean the board, since it is the teacher to do the cleaning. Which pen can I use here that is favorable, that is not there? I think this. So literally this is what we are talking about. So this is our neuron, okay, and the neuron synapses with another neuron, okay, and also this one also synapses with another neuron, and also this one synapses same until when you reach the brain. But however, if we magnify just this part, this one, that point of connection, okay, that's what we call a synapse, and this is what we shall see. Okay, that's what we shall see, where we shall see a swelling, and this swelling is called a presynaptic knob. Knob means a swelling. Okay, but inside that swelling, then you have there some sacs, okay, called vesicles, and these sacs do contain a neurotransmitter substance. You can see this neurotransmitter substance, this one, okay, and then you can see that you have membranes, okay, there is this one. So we are going to see the process of impulse transmission. Then there is this space, the whole of this space is what we call a synaptic cleft. Now this membrane here that is behind the synaptic cleft is the one that is called a presynaptic membrane, this one. Then this one that is just away, this is the cleft, but this one of the postsynaptic knob is called a postsynaptic membrane. Then within that postsynaptic membrane, it's where you have the ligand-gated channels. Now why are they ligand-gated channels? Because they are opened by ligands. A ligand is that either an atom or groups of atoms or a compound that can bind to the central metal metal ion. Let me say that in chemistry. But here in this case, they are gated channels because they have gates, and those gates are always closed. But they are opened when a ligand, and in this case the ligand is acetylcholine. Okay, you can see when a ligand binds, okay, to the receptor. So when that ligand binds to the receptor, then the gate is opened and there will be an influx of sodium ions and potassium ions. So basically that is a picture of a synapse. So I hope you should be able to internalize that in your mind. Take a peek and see a synapse before we talk about the impulse transmission across a synapse. How an impulse can be evoked, can be transmitted from the presynaptic knob to the postsynaptic knob. That is some good science. You can see this. You can see that. So in your mind, you should be thinking, what is this? Are these ones impulses? What is this? I like this peak. So these are they are the neurotransmitter substances. You can see what is happening. But all this, actually, all this science, right from the generation up to transmission along the axon and along the synapse here, it happens within just milliseconds. Just milliseconds. Okay? Let not the science be that wide and you think it is also. No, it happens within just milliseconds. Because imagine that speed you use to withdraw your arm from a hot object. That speed you use. All this that we have studied and what we are going to study, because we still have a lot of science. We still have to talk about transduction. These receptor organs, sensory organs, the eye, the ears, the skin, and so on. We have to talk about them. So all that one happens within just milliseconds. So now here, the first thing that happens is arrival of an impulse, or when an action potential reaches a terminal. This here, the presynaptic knob. If it reaches in this presynaptic knob, then the final result is the release of the neurotransmitter substance into the synaptic cleft. But there is a lot of science that is embedded within this statement, and you need to know. You need to know. So what happens is, when a neurotransmitter substance reaches here, okay, then it opens the calcium gated channels. Okay, the calcium gated channels. get open. That's why in the above, now it will be wide. Here in this one, you saw that you have calcium gated channels here. Okay? So when an impulse arrives, the calcium gated channels, they get opened. Okay? And there are 5 calcium channels are opened, then calcium ions enter. Okay? From this synaptic cleft, they enter into the presynaptic knob inside. So when the calcium ions enter the presynaptic knob, then a high concentration of calcium ions stimulates the vesicles, okay, to move towards the presynaptic membrane. So when they reach there, they will fuse and release this acetylcholine, which is a neurotransmitter, by exocytosis. So that's why you say that arrival of an impulse, the final result is the release of the neurotransmitter substance into the synaptic cleft. Now, when this transmitter substance is released into the synaptic cleft, the result is astonishing. The result is amazing. Okay? That in direct synaptic transmission, we are going to see what happens, what are direct impulse, direct synapses and indirect synapses. But in direct synaptic transmission, okay, the neurotransmitter substance binds to the ligand gated channels. I hope you saw them up, okay? That protein molecule that has that power, okay? The power which allows the ions, okay, the ions, the sodium ions and the potassium ions. So but it has a gate, like how you see, but it has some receptor site somewhere. So when this neurotransmitter substance is released, it will bind, okay, it will bind to that receptor, and binding of that neurotransmitter substance to the receptor in the postsynaptic membrane here, it opens these ligand gated channels. You open the sodium gated channels, okay, and potassium gated channels, but sodium gated channels first, okay. And so when you open the sodium gated channels, then the sodium ions that are highly concentrated in this synaptic cleft, they will easily diffuse. There will be an influx of sodium ions from the synaptic cleft, okay, into the postsynaptic knob inside. So as a result, the inside of this synaptic knob is going to be depolarized, or the membrane is said to be depolarized, in that the inside will gain a positive charge and the outside membrane will gain a negative charge. Sorry for this, a negative charge. And so if this one, we shall say that you have depolarized a membrane, okay, and an action potential is evoked, okay. Now the excitation, or we shall say that the postsynaptic membrane is said to be excited. Now the excitation of the postsynaptic membrane occurs in two, depending on the type of a synapse. Now we have the excitatory postsynaptic potential. Now this one, binding of the neurotransmitter opens the sodium gated channels, so that you have an influx of sodium ions into the postsynaptic knob. And so the excitation that will be created is an inhibitory, so there is an excitatory, is an action potential, which will be transmitted along this neuron to the brain as a wave of depolarization. But however, there is when a neurotransmitter substance, if a neurotransmitter substance binds to the receptors, okay, then you will open the gated channels for potassium and chloride ions. You will open Yes, the gated channels for potassium and chloride ions, or sometimes potassium ions or sometimes chloride ions. Now if you open the gated channels for potassium ions, there will be an outflux of potassium ions. Remember, I have more potassium ions inside. Okay, so the potassium ions will come from inside to the outside. Okay, so if potassium ions come out, then the inside will be highly negative than normal. Okay, and that one we call it hyperpolarization. In other words, the membrane is going to be hyperpolarizing. Similarly, when you open the chloride gated channels, okay, then the chloride ions will diffuse from the synaptic cleft into the postsynaptic membrane. Is that okay? So as a result, the inside will be highly negative than the outside. Then the inside of the postsynaptic membrane will be highly negative than the outside, the postsynaptic membrane. So as a result, it will be hyperpolarized. And so that hyperpolarization creates an inhibitory postsynaptic potential, okay, which now inhibits, or depending on the type of a synapse. If the synapse works by inhibiting, then you will inhibit, okay, and the impulse won't be evoked. But if the impulse is to be continuous, then you will end up exciting, okay, it will end up exciting that synapse and the wave of depolarization is evoked. So it is basically as simple as that. Now, after an action potential has been evoked, this neurotransmitter substance cannot stay, okay, it cannot stay. What happens? It has to be broken down by the enzymes. Which enzyme? It is called acetylcholine sterase. Acetylcholine sterase. So it is that enzyme which breaks down this acetylcholine, and so when you break it down, you release it from the receptors. And so if you release it from the receptors, okay, then these gated channels, they get close again, and if they close again, then you will no longer transmit sodium ions inside, and so everything restores back to normal. So it is basically as simple as this. This is what we've been talking about. So the reason why I put different things is we have people who understand by seeing a picture, or we have people who can understand by listening. So I try to accommodate different kinds of people. So basically this is what we are talking about. So this is our presynaptic knob, and this is our postsynaptic knob. Okay, so this is our synaptic cleft. So when an impulse arrives, okay, and they are here we can read. If action potential arrives near the synaptic cleft, this space, you open the voltage gated channels for calcium ions, okay, and so calcium ions enter the presynaptic membrane. Are you seeing them? Yeah, that's pretty good. That's pretty good. So now if calcium ions enter, then what will happen? It will stimulate these vesicles, these ones, if they are all this side, to move and fuse with a membrane to release their acetylcholine, their neurotransmitter substance. So the synaptic vesicles move and fuse with the presynaptic membrane to release their neurotransmitter substance. Are we okay at that stage? Yeah, that's pretty good. Then what happens when you release the neurotransmitter substance? Then it's number 4 happens. The neurotransmitter substance binds to these gated channels. there is some specific site on these the ligand gated channels. Now the neurotransmitter acts as a ligand, okay? Acts as a ligand. So the neurotransmitter substance binds on the ligand, binds on the site on this ligand gated channel to open up the gate. So you can see this one is bound and it's open, but this one is not yet bound and it is still closed. Okay? And so when you bind and you open, there will be an influx. So this synaptic cleft is full of sodium ions. Okay? So when you open, like how you've opened here, there will be an influx of sodium ions. Okay? Into this postsynaptic membrane or postsynaptic knob. So as a result, ion channels in the postsynaptic membrane open when the neurotransmitter binds. There will be a flow of ions which cause change in postsynaptic cell potential. Okay? So if it is an excitatory postsynaptic membrane, then you open the sodium gated channels and there will be an influx of sodium ions. Okay? And so the membrane is going to be depolarized and it is evoked as a wave of depolarization throughout the entire neuron. Pretty good. But if it is inhibitory, excitatory, synapse, then binding of the neurotransmitter opens the potassium gated channels or the chloride gated channels. And so instead of exciting, you hyperpolarize a membrane. And so if you hyperpolarize, then you inhibit the impulse from being evoked. But this neurotransmitter substance cannot stay. It can't stay. Otherwise, if it stays, then you die of paralysis. You can read about, okay, I promise to talk about it, how pesticides work. I'll talk about it. Because you never know anything. Biology is about application, so they can ask anything. So I'll talk about it. So, there is an enzyme within this space, the synaptic cleft, okay, called acetylcholine sterase. Now that enzyme, it breaks down the acetylcholine into acetyl. From chemistry, you know that acetyl is just CH3, CH2. So into an acetyl group, okay, acetyl group and the choline group. Choline you can find out, however you don't need it. So it breaks down the acetylcholine into acetyl and choline groups. Now these two, once you break down the acetylcholine, then it is detached from this point, okay, released back to the synaptic cleft, fuses back with the vesicles, okay, or absorbed, fuses back with the vesicles, and then absorbed back into this knob to wait for another impulse. Okay? Yes. Basically, that's how it is. That's a summary of impulse transmission. Now, let me talk about the pesticides. Now, most of the pesticides, their target organ, not organ, but anyway, their target site is this synapse. And they most of them target the acetylcholine, which is the enzyme that breaks down this. And did I say that they target acetylcholine? No, they target acetylcholine sterase, which is the enzyme, okay, that breaks down this acetylcholine into acetyl and choline, so that you detach it from this ligand gated channel, okay, so that it closes again. Now, if you inhibit that enzyme, okay, then there is no way how this ligand, how this acetylcholine is going to be broken down. And so, if you don't break down the acetylcholine, then the membrane, the voltage, sorry, the ligand-gated channels, they will remain open. And so if they remain open, there will be a continuous influx of sodium ions. Continuous influx of sodium ions. So as a result, there will be a continuous stimulation. There will be, this membrane will stay depolarized, okay? Which will continuously evoke an action potential that will be transmitted to the brain, and so the brain cells, they will end up that continuous stimulation, continuous stimulation, it will end up damaging the brain cells, okay? And so the organism ends up dying. For instance, if you spray cockroaches, the cockroach, there are stages. There is conversion, anyway, seizures. There is that chitengo, in quotes. So what is bringing the seizures? What is bringing the conversion? It is that continuous stimulation to the brain cells. Okay? So that brain cells end up damaged, and so the organism dies. Okay? So that's how basically pesticides work. And in case you find it somewhere, just think about the synapse and the acetylcholine esterase and the function of acetylcholine. Okay? Yes. I'm attending to your questions now. Now, along the side, remember, from here, you can see that different synapses are formed. They are formed from different dendrites. You can see. You have this, 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 you have this. Now, these action potentials that are produced, okay, by a single synapse, okay, or the action potential that is produced by a single synapse is usually called a graded potential. And usually these graded potential are not enough to exceed the threshold value of a given neuron. But because there are different synapses that are formed by a given neuron, okay? In other words, a neuron forms different synaptic connections, as you are seeing, okay? As you are seeing. Good. Then, these different graded potentials that are usually small, that are usually below the threshold value, they add up to bring about one potential that is enough, that is above the threshold value, that can now be transmitted along this neuron. So that's why you should ask yourself, why does it need to have these very many branches? The reason why, it is summation, and that's what we are going to talk about today. Now, in summation, it is you add up different graded potentials so that you come up with a strong action potential that is way above, sorry, I was taking some water, that is way above the action potential, which can now be evoked as an impulse. So since most neurons have many synapses on their dendrites and cell body, then a single excitatory postsynaptic potential is usually too small to trigger an action potential, okay, in a postsynaptic neuron. That's what it means. Oh my god, sorry for that. This is too far. This is what it means. What is built here is excitatory postsynaptic potential. But a single like this from just one synapse is usually not enough to build an action potential, to form an action potential. So, but the good news is, like how you can see it here, just if you consider this one synapse, okay, a single, this is stimulation number one. Let me get some different pen. I think this one, it's not there. This is stimulation number one, and this will be the excitatory postsynaptic potential that is built up here, E1, and it is way below the threshold value. Now if you wait for some time and you stimulate again, just at the single synapse, still another excitatory postsynaptic potential that is built, it will be way below, okay, the threshold value. So by all or nothing rule, you cannot evoke any nerve impulse. But the good news is, if two excitatory postsynaptic potential are produced rapidly in a succession, like, let me wrap, like you have this stimulation, this is stimulation one, and within that very minute, not even minute, within that very millisecond, you give another stimulation. So they are two in one, okay? It is just rapid and in a single succession, okay? Then you'll be able to produce an excitatory postsynaptic potential, okay, that will be way above the threshold value. And we have said that once it reaches the threshold value, then a full-sized action potential will be evoked. Is that okay? And that type of summation is temporal summation. Now with temporal summation, here you make synaptic junctions with one neuron, okay? However, you stimulate two or more stimuli, you stimulate rapidly, but in succession manner, like, okay? Then it is going to be perceived as one stimulus, which is evoked as a full-sized potential. So the action potential will be able to be evoked or stimulated or transmitted along the axon. And we shall find this when we are studying the eye, okay? When we are talking about the eye and photoreception within the eye, it is where it is prominent. We shall talk about, okay, we shall see. Don't worry about it, we shall see. So basically, that's what we are talking about. So this is the first stimulation, okay? This is your cell body, and there will be the first stimulation, and within the same millisecond, there is another stimulation. So they are two in one, okay? So there will be an E1 that will be built, okay? That is way below, but within that very minute, you stimulate again, so you produce another E1 that will build up way above the threshold value, and a full-sized action potential is evoked. So I hope you get the difference between temporal summation and spatial summation, like what we are coming, what is coming. Now in spatial summation here, you make synaptic connections, synaptic junctions with different synapses, okay? Different synapses, like how you see here. So you have this, you also have this, you also have this, you also have this. Now, when all these ones are stimulated at the same time, the effect, the excitatory postsynaptic potential of one synapse might not be enough, okay, to evoke an action potential. Might be way below, okay, the threshold value. But when the two add up, you stimulate this, you stimulate this. When the two add up, okay, then you bring about an effect that will be enough to evoke an full-size action potential. And the impulse will be transmitted along that axon. It was direct synaptic transmission, where the synapse, so that the neurotransmitter substance binds, and when it binds, it opens the gated channels, the ligand gated channels for sodium ions, and there will be an influx of sodium ions. Okay, that one is with direct synaptic transmission. But now with indirect, now here binding of a neurotransmitter substance, okay, actually doesn't bind anywhere on the ligand gated channel or on the ion channel. It doesn't open the influx of sodium ions. It doesn't lead to depolarization of a membrane. Okay? There is totally no depolarization here. And you don't open the ligand gated channels. But what does it do? It induces a slow response, okay, to a given stimulus. In that, when it binds, then it induces a cascade effect within a cell, within a gland. Actually, this one is specific with glands. Actually, not specific, but it is more of glands. Okay? That when a ligand-gated channel binds, so that when a neurotransmitter binds, okay, to the protein receptor within a cell, okay, then it induces a cascade effect, okay, which will bring about a slow response, but the slow response will bring about a long-lasting effect. If, for example, you want to produce a hormone, maybe adrenaline hormone, you produce a nerve impulse which will bind to the receptors within the adrenomedullary gland, and instead of inducing the depolarization, now here you induce a cascade effect which will lead into the production of a given protein, a given hormone. Now the hormone will bring about the long-lasting effects. However, it is slow. So this binding instead activates a signal transduction pathway. I hope you still remember, because I taught this twice. I first taught it before plants, and again I taught it when we were studying hormones. So it induces a signal transduction pathway that involves use of different secondary messengers, okay, in a postsynaptic cell, okay. Now these second messengers will finally lead to the production of a slowly but long-lasting effect. So this is basically what I'm talking about. So you have a receptor, and this is the extracellular matrix, and this is the intercellular matrix. So you have a receptor where your neurotransmitter substance is going to bind. Now binding of this neurotransmitter substance induces this G-coupled receptor, this G protein, okay, to undergo some configurational changes, okay, which will induce activation of second messengers within the cytoplasm that will activate a gene that is specific to the stimulus, so that now that gene leads to production of protein molecules, okay, that will respond accordingly to the situation at hand. So this one, it is indirect synaptic transmission. Okay, that's pretty good. But with the direct, it leads to formation of the excitatory or inhibitory postsynaptic potential. But this one leads to a signal transduction pathway. Is that okay? I hope it is okay now. I hope it is okay now. So basically that's where we are going to stop today. And we are going to talk about the neuromuscular junction and we shall stop here. But before the neuromuscular junction, and we call it that. Now these ones, they shouldn't confuse you. These ones, here the neuron, the nerve fiber makes synaptic connections with the muscle cells. Okay? So here there is the terminal dendrite. Let me get some pen. The terminal dendrite of a motor nerve. I hope you still remember what a motor nerve is. Makes synaptic connections with a muscle fiber. Okay? So the other one we saw it was a gland. Now here you are making synaptic connections with a muscle fiber. So also this one is okay, we shall see. Now at this nerve muscle junction, the membrane of the muscle fiber is modified to form a motor end plate. Okay? To which the dendrites must now be attached. And you can see an illustration from here. Okay? Maybe let me try to magnify for us so you can see an illustration. So basically that's how it is. So if this is your motor neuron, the motor neuron, it makes synaptic connections, okay, with a nerve cell. Now that one is called a neuromuscular junction. Now the details you study, the details, how it brings about contraction in locomotion. Okay? You need this science for you to study, for you to understand locomotion. But the concept is the same. Okay? That when an impulse arrives, like how you see, maybe we can magnify. Oh, sorry. When an impulse arrives, like how you see, okay, then you open the calcium gated channels, okay, calcium enters, and then you release your neurotransmitter substance. Now the neurotransmitter substance, it is the one that will bind to these protein receptors, okay, to these ligand gated channels, okay, within the muscle. Are we together? And now this one, the ligand gated channels, they will now allow the influx of calcium ions into the muscle cell, the contraction unit, which will bring about the muscle contraction. Okay? And now the process of muscle contraction, you can read about the sliding theory of muscular contraction. And the science. Now from here, that's where I'll stop. The next, you study it in locomotion. Or if we study locomotion together, we shall talk about it. It will just be a reminder. Okay? So we have now transmitted our impulse from the point of perception to the brain, to the glands, up to the muscle. So when an impulse arrives, you can read through at the nerve muscle junction, then acetylcholine is discharged from the synaptic vesicle into the synaptic cleft, this space. And then the acetylcholine diffuses across the gap and depolarizes the muscle and plate. Okay, and so if you record, then you can see that different action potentials are built up, which result into the contraction of the muscle cell. So it is as simple as that.